Welcome to host Pathogen Interactions course from NPTEL. This is week's 11 live session. I am the course TA Sumati Raviraj from University of Hyderabad, and the course instructor is Professor Himanshu Kumar from ISER. So this week the topics that were covered were introduction to bacterial infection, virulence factor during bacterial infection, particularly tuberculosis, in 3 different parts. So the videos are available on the NPTEL website course page. Please go through those, and this presentation or this session is to help you solve the assignment questions. So let's go into the topics where we will be discussing the main topic of each question, so that a basic understanding of the question is given, and also it will help you answer the question. So the first was regarding endotoxins. So endotoxins are something which are present in bacteria, especially the gram-negative bacteria. So the origin and the structural details regarding this. The source is only in gram-negative bacteria, and the location is, that is where it is present, is it on the outer membrane of the bacteria, that is the cell wall, and the composition of it is usually lipid, polysaccharide, and O antigen. So lipid A is a toxic component, core polysaccharide is present, and O antigen, which is variable and it can be antigenic in nature and it involved in evoking the immune response. Then the biological properties, that is coming to the toxicity of it, it is moderately toxic, much less than exotoxins, and the mechanism is it is released when the bacteria dies or when the bacteria is lysed, that is upon the death of bacteria, when the lysis or degradation of the bacteria occur, because it's present in the outer membrane of the bacteria, it has to be lysed and it will be released. Then it activates the immune cells, namely the macrophages, dendritic cells, via toll-like receptor 4, that is TLR4. The main effects, like the physiological symptoms when exposed to such endotoxins, is it is pyrogenic, that is it invokes fever in the system, and it is inflammatory in nature, so there is inflammation in the body. Then vasodilation occurs, hypotension occurs, and at higher levels, that is if the host is exposed to high levels of endotoxin, it can also lead to septic shock. Then the physical and chemical characteristics of it, regarding the heat stability, it is very stable, that is it can tolerate high temperatures, and it can also resist boiling and autoclaving. Then it cannot be converted into toxoids like exotoxins, and it is not secreted usually like exotoxins. It is released upon bacterial lysis or death. Then it is non-enzymatic, and it is also not tissue specific. So this is some of the characteristics. And how to detect the exposure to endotoxin or the presence of endotoxin is LAL test, that is Limulus amoebocyte lysate. So sensitivity test, and sensitive test used to detect endotoxins, and especially in the pharmaceuticals and injectable drugs. So it's highly sensitive, and it helps in detecting the levels of endotoxins. So the basic will again come will further be talking about the differences between endotoxin and exotoxin, but just a basic understanding of it, so that you can easily remember if you compare both of them. So the source for endotoxin is gram negative only, that is gram negative bacteria, whereas the source for exotoxin is both gram positive and gram negative. Then the location is endotoxin is outer membrane, exotoxin is secreted by live bacteria. It is, since it is in the outer membrane, it can only be released upon the death of the bacteria or due to lysis of the bacteria. Then heat stability, endotoxin is highly stable and exotoxin is usually heat labile, that is, it's not very stable. And toxicity is endotoxin is moderately toxic and exotoxin is often uh, very high toxicity is seen on exposure of exotoxins. Then toxoid conversion, that is whether it can be used to convert into toxoids. It is a no for endotoxin, that we also saw here that it cannot be converted into toxoids, but exotoxin can be converted into toxoids. Then immune response, it is immunogenic, but however, endotoxin is weakly immunogenic and exotoxin is strongly immunogenic. So these are the key differences or basic differences between exotoxin and endotoxin. Then coming to the next topic, it is regarding the granuloma formation in Mycobacterium tuberculosis. So in granuloma, that is formed during the MTB infection, several immune cells come together to contain the bacteria. So this is what a granuloma is, where several immune cells, you can see here, they come to contain, that is to uh, come limit the bacteria at a particular place. They come and surround it like this, and this will also help in prevention of spreading of the bacteria into different places. So in such cases it becomes a granuloma. So the major objective is to contain the bacteria. So the core cells, what are all the cells that are present? You can see the T cell, B cell, neutrophil, NK cell, epithelioid, macrophage, foam cells, dendritic cell, giant cells. So there are several types of immune cells that are involved. So coming to particular regions, in which region which cells are first present. So in the core region, that is in the central region, you can see here surrounding immediately the bacteria, the infected macrophages. So they engulf the MTB but often cannot kill it. So though macrophages are phagocytic cells, they engulf it but they cannot kill the MTB. So they serve as a reservoir for the bacteria. So they engulf it and they store it. So it becomes a reserve of the bacteria since they are unable to phagocytose. Then epithelioid cells. So differentiated macrophages with enhanced secretory functions. So these are also differentiated macrophages with secretory function and they have more epithelial-like appearance. Then also multinucleated giant cells. You can see here multinucleated giant cells, the Langhans giant cells. So they are formed by the fusion of macrophages. They contain multiple nuclei because they are formed by the fusion and they often are arranged in a horseshoe like pattern. So if you see a horseshoe, it will be like that. So the arrangement is in that pattern. Then this is the central region, that is the core region, where the macrophages, mainly epithelioid and giant cells, are present. You can see here, these are the macrophages, these are the epithelioid macrophages, and these are the giant cells present in the core area. Then in the peripheral region, you can see here we have the B cells, NK cells, T cells, etc. So here we have the T lymphocytes, that is the major immune cells, mainly the CD4 plus T cells. So they coordinate the immune response, release IFN gamma to activate the macrophages. Then CD8 plus cells, CD8 plus T cells, they are also present and they kill the infected macrophages and help contain the infection. Then we also have B cells, so found at the periphery and may form follicles and involved in antigen presentation. As you know, they are APC cells, so antigen presentation and also antibody production, because B cells humoral in immunity. So though they are not central in the control. Then we also have dendritic cells, so they present the antigens to the T cells. They are also APC, then help initiate the adaptive immunity. Then we also have fibroblasts in more chronic and organized granulomas, like in highly developed granulomas at later stages in chronic conditions. We also have fibroblasts present. So they lay down the collagen and contribute to fibrosis and granuloma structure. So basically, granulomas aim to contain the infection, not necessarily to eliminate it. So caseous necrosis. So this is something like a cheese-like central area. So this may develop due to immune-mediated tissue damage. And in latent tuberculosis, the granuloma can remain stable for several years. So there is latent and then there is active TB. So in latent TB, even though you don't have the symptoms or you don't have the effect much, the granulomas are still present and are stable for years. And in active TB, breakdown of granulomas can release the bacteria and spread the infection further. So this is the difference between this and also the different types of cells that are present in the granuloma. Then comes the next one, that is extracellular bacterial infections. So extracellular bacteria are pathogens that do not invade the host cells. So basically they do not enter the host cells, they do not use the host cells, they are extracellular in nature, but instead multiply outside the cells in the body fluids, tissues, or in the mucosal surface. So they are usually present, or they reproduce or multiply in the interstitial tissues, in the fluids that are present, that is your mucosal surface or other body fluids, depending on the type of the extracellular bacteria. So the immune response to extracellular bacteria, coming to the innate immunity, we need the complement system, especially the classical and alternative pathways are activated, and then neutrophils to phagocytose the bacteria, macrophages also to phagocyte, engulf and phagocytose the bacteria. Then inflammatory cytokines, pro-inflammatory cytokines are produced. Then in case of adaptive immunity, humoral antibody mediated immunity is primary here, and the key players are Th2 cells that stimulate the B cells, and the B cells from the plasma cells, they produce the B plasma cells, they produce the antibodies, your IgG, IgA, IgM, and then majorly they function to neutralize, opsonize, and in agglutination, as well as in activation of the complement system. So neutralization, it blocks the bacterial toxins and reduces the adherence, and opsonization, it tags the bacteria for phagocytosis, and in case of complement activation, your classical and alternative pathway eventually lead to the MAC formation and bacterial lysis occurs. And agglutination, clumps of bacteria are made, that is they agglutinate together, agglutinates are formed, so that they are easier for recognition and clearance. So this is how your innate and adaptive immunity response to the extracellular bacteria that infects the host. In case of disease mechanism of the extracellular bacteria, Toxin production is seen. Example, in case of cholera, tetanus, etc., we have the toxins, and inflammation is present in the body due to the toxins and the bacteria, uh, leading to tissue damage because excessive pro-inflammatory cytokines are released, and then immune evasion by capsule formation or biofilm formation or antigenic variation. So the bacteria, pathogenic bacteria, also adapt or evolve sometimes, or they have evolved, few of them, to escape the immune response. So in such cases, few of the bacteria, they have capsules. They form capsules which are not easily destroyed. They cannot be they're very stable, they are heat stable, pH stable, so they cannot be excluded or attacked. And many bacteria form biofilm. So when biofilm is also formed, they can have communication easily. They can change, or they can, they can divide the functions. They can divide labor between them to effectively establish the infection. So in such cases also, they are difficult to eliminate. And of course, there is antigenic variation. So if there is always change in the antigen, if it is changing the genetic material, or it is having multiple antigenic points, or it often evolves to present a different antigenic region or epitope, then there is always a secondary infection, or it can always, even after prior infection, the immune system will not be able to, provide protection against it the next time. Then, so the main key players, what we were discussing for this is the response of Th2, cell mediated immunity, and complement system activation. So Th2 cells promote B cell activation, as we discussed, and help in antibody production, especially the IgG, IgA, and IgE. So it is important for fighting extracellular pathogens, including the bacteria and other parasites as well. Then Th2 cytokines like interleukins 4, 5, 13 help in activation of B cells, promoting class switching, recruiting eosinophils. So for more of parasitic pathogens, parasites, or you know these eosinophils play more important role. Multicellular parasites. Then complement system activation, part of it is part of the innate immunity, but it works with antibodies because we need the antigen antibody complexes, especially the IgM and IgG are the key activators of the complement system. So they finally help in formation of the MAC complex, if you remember from the previous sessions. So it finally creates a pore in the bacteria and helps in bacterial lysis and opsonize the bacteria, that is tag the bacteria for phagocytosis, then promote inflammation and recruit immune cells. So this is the major two responses of the host system against extracellular bacteria. Next is pyrazinamide, the mechanism of action. So it is pro-drug activation. So pyrazinamide is an inactive pro-drug. It is converted inside the MTB by bacterial enzyme pyrazinamidase, encoded by the PNCA gene, into its active form. So since it's a pro-drug, it has to be enzyme acted by an enzyme, and it has to be converted into its active form. So pyrazinoic acid or POA is the active form of it. Just a second. think it got disconnected. Then yeah, so for this drug, acidic environment is key. That is, POA works best under acidic conditions, specifically the acidic pH found in phagolysosomes inside the macrophages. So this environment favors the conversion of POA to its protonated form, that is HPOA. So passive diffusion of HPOA into the mycobacterial cell occurs. Then disruption of membrane transport occurs. So this is the major mode of action. Once inside the bacterial cell, the HPOA, that is the protonated POA, becomes releases a proton. So it releases H plus and then it becomes negatively charged, and then it results in POA minus, that is negatively charged POA, which then gets trapped inside the bacterial cytoplasm. So basically it disrupts the membrane potential. So accumulation of negatively charged POA inside the bacterium, this disrupts the membrane potential, that is the electrochemical gradient across the membrane, and then it also interferes with the membrane transport functions. Then especially in case of nutrient uptake and proton motive force. So when the gradient is disrupted, the proton motive force is disrupted, then nutrient uptake is disrupted, then ATP production will be disrupted, etc. Then this leads to acidification of the cytoplasm because more POA, negatively charged POA, is accumulating and destabilizes membrane integrity. So particularly in non-replicating dormant bacteria. This ultimately causes loss of bacterial energy production, disruption of homeostasis, death of bacterium, especially in latent or dormant states where other drugs are less effective. So the primary anti-TB activity of PZA or POA comes from accumulation of pyrazinoic acid inside the cell, that is a negatively charged POA, and disruption of the membrane energetics uh, due to this. This especially effective against dormant bacilli, a unique feature of POA or PZA among TB drugs. So uh, yeah, so this drug is a pro drug. As we discussed, it's activated by PNCA, that is pyrazinamidase, and leads to its active form, that is POA, and active in acidic environment, and main function is disrupting the membrane transport and energetics, effective against dormant slow growing TB bacilli. Just a second, I will be back. Yeah, so this was regarding the PZA, that is the pyrazinamide. Now coming to the Bacillus anthracis. So this is the anthrax bacteria. So Bacillus anthracis, excuse me. So Bacillus anthracis is a gram-positive bacteria rod shaped, and it is a spore forming bacteria. Yeah, so it's a spore forming bacteria that causes anthrax disease, and it is a zoonotic disease affecting both animals and humans. So you can see here the labeled Bacillus anthracis. This is the rod-shaped bacteria, and it is sporulating, and it has an endospore in the center, and you can see its genome and then other cellular organelles present, and a spore membrane present around the spore, and this is how it looks upon staining. So you can see here, this is the bacteria, and in the center you can see the spores in it. So it's a rod shaped gram negative bacteria with endospores present. So basic description, and it is an aerobic bacteria, and sometimes it can also be facultative anaerobe. Then spores are highly resistant spores, and it is non-motile, and the capsule is made up of poly-D-glutamic acid. The diseases in humans: cutaneous anthrax, inhalational anthrax, gastrointestinal anthrax, injectional anthrax, etc. So cutaneous is how it transmits via skin, most common, and it is least lethal. Then inhalational anthrax via inhalation of the spores. So this is highly lethal and it has high mortality rate. And then gastrointestinal anthrax via ingestion of contaminated meat. So the spores can be present in contaminated meat. And injectional anthrax, it's very rare, but seen in IV drugs uses. So sharing common needles and not using sterile needles can also lead to such kind of transmission. Then virulence factors. B. anthracis has two main plasmids that encodes its major virulence factors. That is the plasmid PXO1, that is anthrax toxin components are coded by this. So protective antigen PA, edema factor EF, and lethal factor LF. So protective antigen binds to host cells and allows entry of other toxin components. And edema factor and adenylate cyclase, it increases CAMP and causes edema. Then zinc, lethal factor is zinc metalloprotease. It cleaves the MAPK kinases, and MAPK pathway is activated, disrupted, and it leads to cell death. So PAEF together is edema toxin, PALF is lethal toxin. So both of them require PA. And then other than this, for the capsule, we have plasmid PXO2. So this encodes for poly-D-glutamic acid of the capsule, and it prevents phagocytosis, helping the bacterium invade the immune system, evade or escape the immune system. So these are the major two virulence factors. Then prevention and treatment: vaccination. So vaccines are available for high risk individuals, and antibiotics like ciprofloxacin, doxycycline or penicillin are early treatment, and antitoxins for toxin neutralization in severe cases are done. So, vaccine, antibiotics, antitoxins. Then, particularly about Mycobacterium tuberculosis, it is why it has to be studied so much is because it presents a major global health challenge, primarily because of its ability to develop resistance to many antibiotics. That is, multi-drug resistance is seen in case of it. So, effective treatment cannot be done. So, it makes the treatment difficult and prolonged. Multi-drug resistance in case of MTB can become resistant to isoniazid and rifampicin, the two most effective first-line drugs. Then treating MDR TB, that is multi-drug resistance in TB, requires second-line drugs which are less effective and more toxic and much more expensive, and the treatment time is also prolonged. It needs to be taken for about 18 to 24 months or even longer depending on the severity of the case. And extensively the drug resistant TB, that is resistant to the first line of drugs plus key second line drugs. So this is highly drug resistant or extensive drug resistance in case of few of them. So this is even harder to treat because here at least the second line of drugs can be used, but if they again get resistance for the second line of drugs, then the mortality rates definitely increase. So the major challenges are the intracellular survival. The impact is it escapes the immune system, so immune evasion is seen because of the capsule. And then latency, it is a long-term reservoir and is present as a dormant bacterium, and risk of reactivation is seen even after the treatment is done. So long-term treatment duration, that is due to the poor adherence and resistance risk. And then drug resistance, so because of this it's harder to treat and it's more costlier and time consuming. And airborne spread, so airborne spread is highly transmissible because you can avoid contaminated food, or you can be you can take precautions against use of needles and all these things, but if it is airborne you cannot prevent it in a very efficient way. So it is highly transmissible, and especially in crowded areas. So weak vaccine, so it is of limited prevention. And then HIV co-infection is a major uh, complication of this. So when there is HIV co-infection along with TB, it increases the TB burden and definitely increases the mortality. So drug resistant TB, the multi-drug resistant TB manifests resistance to both isoniazid and rifampicin. Globally, around 3.4% of the new TB patients and 20% of the patients with a history of preceding medication for TB were identified as multi-drug resistant to TB. And then we also have extensively drug resistant TB, that is the XTR TB. So MDR strains that offer additional resistance along with the first line drugs, they have resistance to the key second line drugs like fluoroquinolone, so plus at least one of the following uh, linezolid or betaquilin. So these are XDRs, that is extensively drug resistant. And drug resistance can be caused by incorrect prescribing of the medications. It can be due to irregular supply of the drug, incorrect usage, not completion of the course, patient non-adherence, low grade quality of drugs, etc. may also contribute to this drug resistance. So this is about the TB and its drug resistance. So coming up to the exotoxin versus the endotoxin. So what is first an exotoxin? What is first an endotoxin? So exotoxin are proteins actively secreted by bacteria often act on specific targets like nerves or intestines. And endotoxins, however, are part of the membrane and are not secreted by the bacteria, and they are present only in the gram-negative bacteria, specifically the lipid A portion of the LPS layer, and it is only released when the bacteria dies or the bacteria is lysed. So this is the major definition of exotoxin and an endotoxin. So coming to the comparison of it, production of the toxins. So it is produced by the exotoxin is produced by both gram positive and gram negative bacteria, whereas the endotoxin is present only in the gram negative bacteria. So it is located, the exotoxin is located or secreted outside the bacterial cell wall. However, the endotoxin is a part of the outer membrane, that is the lipopolysaccharide layer. And exotoxin are mostly proteins in nature, and endotoxin is, however, the lipid part of the LPS. And exotoxins are heat labile, that is, they can be inactivated by heat. However, the endotoxins are highly heat stable. And exotoxins are highly toxic even in small amounts. Endotoxins are, however, less toxic, effects only seen in higher concentrations. Coming to immunogenicity, exotoxins are strong immunogens, that is, it stimulates antibody production, and endotoxins are weak immunogens, that is, it provides only poor antibody response. Toxoid formation, exotoxins are, yes, can be converted into toxoids for vaccines. Endotoxins cannot be used as toxoids for vaccines. Then effect specificity. So exotoxins have specific effects that is particular to the type of protein it is. It can be either a neurotoxin, it can be an enterotoxin, so it specifically attacks the nerves or the intestines. Endotoxins are, however, non-specific, and they can only cause basic symptoms like fever, shock, and inflammation. But however, they can also be lethal. In case of higher concentration, it can lead to septic shocks. Examples of exotoxins are botulinum toxin, that is the Clostridium botulinum toxin, diphtheria toxin, and cholera toxin. Endotoxins are the LPA, LPS lipidae, and these are present in most of the gram-negative bacteria. All the gram-negative bacteria have LPS layers, so it's present in them. So, like even the host residential bacteria like E. coli can also sometimes be an opportunistic pathogen and become an can release endotoxin. So other than that, salmonella, other gram-negative bacteria, all of them have the LPS layer. So they all have lipid A, which can be released upon their death or lysis. These are the major differences between the endotoxins and the exotoxins. Okay. Then comes the clinical relevance of it. So exotoxins cause diseases like tetanus, botulism, cholera. Endotoxins cause endotoxic shock, that is the septic shock, fever, DIC, inflammation, and sepsis, and is only by the gram-negative bacteria. So exotoxins are proteins produced inside the pathogenic bacteria which are secreted into the surrounding medium. You can see the alive bacteria here, which continuously produces or releases, secretes the exotoxins. The exotoxins then go and attack the target cells specifically and damage them. Endotoxins are either by the natural death of the bacteria or when it is phagocytosed by the macrophages or any phagocytic cell and it is lysed. So then these endotoxins are released and then they enter into the bloodstream, and they are mostly in the are non-specific in nature, so they can go or they can have their effects anywhere in the body, and it is the effects are seen as a whole in the system. Then next is the main limitation of Montauk's test. Uh, it cannot distinguish between the active TB, latent TB, or past exposure, including from BCG vaccination. So Montauk's test, the limitation is it cannot say which, like, is it an active TB? Is the infection current, or it is dormant? That is, it is latent TB, or is it due to a past exposure, which of some vaccination, that is the BCG vaccine? So all these cannot be differentiated in Montauk's test. So this becomes an issue in diagnosis and then later for treatment. So then it results in false positives and false negatives. So a person may test positive due to previous BCG vaccination. So the test might show the person is infected with TB, that is, it is positive, but that could also be because of the vaccination. So the exposure to non-tuberculosis mycobacterium can also give positive, and environmental bacteria that aren't actually MTB. So these kind of false positives can also be result, and these results don't necessarily mean the person has an active TB. Then there is also chances of false negative. So a person may test negative even though he is already infected and has the TB bacterium present in him. So this could be because of the weakened immune system, because it is an immune response that is checked. So this could also be a result of secondary complications or secondary issues already present in the person, like he can be a HIV positive person. So the immune system is not very efficient, and malnutrition can also result in weakened immune system. Immunosuppressive therapy, the person might be undergoing some drug treatment, some immunosuppressives might be given which might have suppressed the immune system. So in such cases you might have false negative, wherein in reality the person is actually infected with TB. And also very recent infection, like if the infection is very recent, then also you can get a false negative, because it has a window period where the enough number of bacteria should be there to invoke the immune response, or the immune response has to be activated. So the window period for testing for TB is 2 to 8 weeks. So anywhere between, depending on the host system, depending on the amount of bacteria that has been infected, and many other reasons, the test can show positive anywhere between 2 to 8 weeks. And also incorrect administration or interpretation of the test. So if the test is done, if it's not done properly, also you will not get a positive result. No information on disease activity. So a positive test only means prior sensitization to TB antigens. It does not confirm whether TB is active, latent, or even still present. So as you can see that BCG also shows a positive. So even if you are sensitized to the antigen and do not really have the infection, or whether it's an active state, latent state, it cannot be confirmed. So the alternatives. So alternatives or complementary tests are interferon gamma release assay, that is IGRA, more specific, and especially in case of BCG vaccinated individuals. So most of the people are BCG vaccinated. So in such cases, it's better to go for alternatives. And a chest X-ray definitely, and a sputum testing can diagnose for active TB. So the limitation of Montauk's test, it cannot distinguish between active and latent, and this may result in unnecessary treatment or missed diagnosis. And another is affected by BCG vaccine, so false positives in case of vaccinated individuals, affected by immune suppression, so false negative in case of immunocompromised people. So this is how the test is done. So that is the reason the effective, if the test is not done properly, also it can be false negatives. So the The antigenic derivative of the TB is injected in the derma of the skin. So to see an antigenic reaction or immune reaction like this. So based on the diameter of the pustule or the inflammation that is seen, the diagnosis is made. So in case of negative reaction where it is injected, only a slight redness will be present or complete absence of a tubercle will be present. So there will be nothing really, maybe just a redness due to the injection. In case of a tubercle formation, that is like a pustule, like here you can see clearly a smaller pustule like this, or a bump that is seen on the skin. So in case of that, you have to measure it, and then if it is 2 to 4 millimeter redness without a tubercle, that is without formation of like a pause, without formation of proper pustule, if it is just increased like a swelling, or if it is increased redness in case of diameter, then it is a doubtful reaction, so we cannot really confirm in this. A positive reaction is said when the diameter of the tubercle or the pustule is 4 to 9 millimeter to 15 millimeters. So 4 to 9 millimeters is a weak reaction, and 10 to 15 millimeters is an average reaction, and 15 millimeters is a strong reaction. And then hyperergic reaction is when the diameter is over 17 and you see a proper a pustule formed in the person. So these are how it differs based on the amount of reaction that is seen. Then comes the granuloma formation in tuberculosis. So granuloma formation in tuberculosis is a hallmark of immune response to Mycobacterium tuberculosis. That is, it is the key characteristic of it, and it represents the body's effort to wall off or contain the bacteria, preventing it from spreading. So it basically like surrounds it, contains it so that it doesn't spread. That is the basic actual origin of a granuloma. Why and how granulomas form? So the immune system recognizes the infection but can't completely eliminate the bacteria. So it tries to wall it off. So since it cannot attack it, it cannot eliminate it, it cannot kill it, it thinks at least if I contain it to this particular area, it will not spread more from here. So the macrophages infected with TB recruit other immune cells. The T helper cells activate these macrophages using IF and gamma, improving their ability to kill the bacteria. Then we also have epithelioid cells, like we discussed earlier. These are activated macrophages, and multinucleated giant cells, lymphocytes, form a tight cluster around the infected site. The center often becomes caseous necrotic, that is due to the dead cells in the center, a hallmark of TB granulomas. This structure is called a granuloma, and it helps prevent the spread of the bacteria to the other parts of the lungs or the body. Majorly, it affects the lungs, so it is confined over there. What does it indicate? It's a protective mechanism, not a sign of failure. It shows that, so it's like plan B. So if you cannot kill it, at least hold it in that particular region. It shows that the immune system is engaged in attempting to limit the bacterial growth. It can contain the infection in a latent state, meaning the bacteria are still present but inactive. However, the immune system weakens if or when the immune system weakens, in case of other infections like HIV, or due to age or due to malnutrition, the granuloma can then break down, and it can lead to release of the bacteria and lead to active TB disease. Let me just connect to the charger. So, coming to the clinical insights, latent TB, the granulomas are intact, the person is asymptomatic and not contagious, but there is a risk a latent TB can any time become an active TB because of the reasons discussed before. So, in case of active TB, the granulomas break down, bacteria escape, and symptoms appear. So, major symptoms are cough, fever, weight loss, and the person becomes highly contagious. So, basic features and functions made the granuloma. It is made up of immune cells and it is coordinated immune response. Forms around the TB bacteria because it attempts to isolate it and contain the infection. Center may show necrosis. This reflects the immune attack and the cell death. Sign of immune control, not failure. So the bacteria are held in check often in the latent TB state. This was about the granuloma in case of TB. Then coming to the last question. So here we'll directly discuss the true and false. So this was regarding the bacterial regulation and pathogenicity. So the statements given were bacteria can exist in both free living and pathogenic state. Environmental signals can trigger changes in bacterial growth modes. Co-evolution of bacteria and host immune system affects infection dynamics. Secretory IgA plays a role in preventing bacterial colonization. Surface properties of bacteria do not influence their adherence to the host tissue. The immune system only responds to the presence of exotoxins. All kinds of bacteria can produce endotoxin and exotoxin. Understanding toxin mechanisms is irrelevant for vaccine development. So out of these are the true statements, that is bacteria can exist both free living and in pathogenic state. So there are many opportunistic bacteria, like I said, like E. coli, which can live harmlessly in the environment or even in the gut of humans and other animals, but it becomes pathogenic under certain conditions and it acquires virulence factors or virulence genes and then behaves as a pathogen. Then environmental signals can trigger changes in bacterial growth modes. Yes. So the bacteria responds to various factors like the temperature, the pH, the availability of nutrients, and it switches between planktonic and biofilm modes or activating its virulence genes, etc. So environmental stimulus can trigger changes in them. Then co-evolution of bacteria and the host system affects infection dynamics. Yeah. So host and pathogen always co-evolve. So the bacteria evolve to evade the immune detection. That is, the bacteria evolves in a way to escape whatever attack it is receiving from the immune system, while the host adapts to recognize and fight back. So the host tries to recognize it in various ways and tries to attack it in various ways, and this always uh, keeps happening, and this influences the disease outcomes and resistance pattern. Next is secretory IgA plays a role in preventing bacterial colonization. Yeah. So secretory IgA is found in the mucosal surfaces like the gut, the respiratory tract, uh, and it blocks the bacterial adherence and colonization. So other than these, the other statements were false. That is, the surface properties of bacteria do not influence their adherence to the host tissue. This is false because the surface structures like the pili or the fimbriae and the adhesins and other proteins are crucial for the membrane attachment or the bacterial attachment to the host and to establish the infection. Then next was the immune system only responds to the presence of exotoxin. This is false because we know endotoxins also evoke immune response. The immune system responds to many bacterial components such as the cell wall, the LPS, the peptidoglycan, flagella, endotoxin, exotoxin, and much, much more. And other than this, all kinds of bacteria can produce endotoxins and exotoxins. No. So this is because endotoxins are produced only by only by gram-negative bacteria. That is because they have the LPS layer, which has the lipid A, which is the endotoxin. And exotoxins are secreted proteins are produced by both gram-positive and gram-negative bacteria. So this statement is also false. And the next one is understanding the toxin mechanism is irrelevant for vaccine development. So this is completely false, because only if you understand the toxin mechanism you can make a vaccine, you can develop a vaccine. So many vaccines like the diphtheria, tetanus are based on inactivated toxins that are toxoids. So understanding their basic mechanism or their mode of action, mechanism of action is critical, so that these toxins can be converted into toxoids to give as a vaccine. So out of all these, the correct answers or the correct options are the bacteria can exist in both free living and pathogenic states. Environmental signals can trigger changes in the bacterial growth modes. Co-evolution of bacteria and host immune system affects the infection dynamics. Secretory IgA plays a role in preventing bacterial colonization. So these are all the correct statements and these are all the false statements. So that is it for this session. So we discussed a lot about granuloma, MTB test, its limitations, exotoxin, endotoxins, bacillus anthracis, little bit, PCA drug, and extracellular bacterial pathogens, and that is it. So this was the week 11. So if you have any doubts regarding this session or any other topic, you can always write down to this mail ID or put it up in the course portal. So your queries will be addressed there. So this PPT will be shared and the video will be posted on YouTube. So we can, I think, end the session here. Thank you.